My name is Carolyn Chapman, and I lead the Cell and Gene Therapies project at the MRCT Center. I'm a lead investigator at Brigham and Women's Hospital and member of the faculty at Harvard Medical School. And in this session, I'm going to provide a brief overview of the topic, and then we're going to move to a discussion with our panelists. But before we get started, I wanted to start by thanking MRCT Center colleagues, all of them, but especially those who are part of the Cell and Gene Therapies team, and that's Amina Burbeck, who's program manager, Mina Sheikh, research assistant, Lindsay Henriquez, graphics and information design specialist, and Barbara Beer, our faculty director. So I thought that it would be important to first orient you to the Cell and Gene Therapies project. So the Cell and Gene Therapies project are aligned, the objectives are aligned with the MRCT Center's mission. And so what we're aiming to do is to first identify and characterize ethical, regulatory, and logistical challenges arising in the context of global research and development of cell and gene therapies. And then the second objective is to collaborate using a multi-stakeholder approach to co-develop actionable and practical mechanisms for addressing these challenges to support efficient, safe, and respectful clinical development of cell and gene therapies. So when we started the project about 2 years ago, we had to figure out which specific issue or topic we should dig into first. And we were looking for an issue right at this intersection, of course, cell and gene therapies, these ethical, regulatory, and logistical challenges, and an issue that would fit with the MRCT Center focus or approach, which really is one, in my mind at least, that cannot be solved by any one entity, but really needs a collaborative approach. And so what we chose was long-term follow-up for gene therapies. So let me tell you a little bit about long-term follow-up studies. So they involve extended monitoring of recipients of either investigational or approved gene therapies, including genetically modified cell therapies such as CAR-Ts. And gene therapies are a class of pharmaceutical products which involve the modification, replacement, or addition of genetic material to human cells. And although they have the potential for lasting and durable beneficial health outcomes, often after one dose or administration, they also have the potential to cause delayed adverse events, such as malignancies, immunological reactions, or infections. And because they're still a relatively new class of therapeutics, there's still significant uncertainty about the long-term benefit and risk profile of these products. So therefore, long-term follow-up is really important, and the information from these studies is valuable to many different stakeholder groups, of course, including to patients and participants in the studies and the people who support them, the medical care teams that support them in research and clinical care decision-making. And they're also, of course, important to regulators and sponsors as well to know whether the products are safe and effective. But here's the thing, they fit that criteria of having challenges, right? So because of the length of these sort of unprecedented length of these studies, which can last 5 years, 15 years, and perhaps involve monitoring of patients for a lifetime, they can be really challenging and expensive to design, conduct, and operationalize. And of course, this is challenging for sponsors and academic investigators, as well as the study sites. And not to mention, it's very burdensome on patients in terms of time, expense, and opportunity costs as well. Okay, so now that we had the challenge in hand, in true MRCT Center fashion, we formed a long-term follow-up working group launched in September 2024, which currently has 25 members. And I just want to pause and thank all the members for their support and contributions to this work. We have such a great group, and from the beginning, we set out to build from and off regulatory guidance to develop guidance on best practices and associated tools for the ethical design and conduct of long-term follow-up studies for gene therapies, as well as to hypothesize innovative approaches. And we have monthly meetings as a group, and also communicate in between the meetings. Along the way, we have hosted and held webinars on long-term follow-up to push the conversation forward. This includes 2 webinars in 2025, one on patient-centered long-term follow-up for gene therapies, and another one on innovative approaches. Both of these are on demand, available on demand on our website, as is a webinar that we co-hosted with NYU's Pediatric Gene Therapies and Medical Ethics Group in December of 2024, also available online. So it's really my great pleasure to, on behalf of the MRCT Center team and the working group, to announce that we have a major milestone. On November 4th, we're launching a toolkit for supporting the design, conduct, and reporting of long-term follow-up studies for gene therapies as a draft for public comment. And that day, we're also going to have a webinar to mark the release with a few different panelists, so we'll broaden the conversation even more. So in this section, I just wanted to give you a little preview, a very small taste of things that are in the toolkit, and then I think the panelists are going to talk about other aspects of the toolkit as well, or other topics that are addressed in the toolkit. So first I wanted to talk about the structure of the toolkit. So the heart of the toolkit really is 3 different sections. One is a guiding principles section, another is considerations and recommendations, and then another is a looking forward section. And then there are additional resources, including description about different types of long-term follow-up studies, flowcharts, key design elements of long-term follow-up studies that have been implemented by FDA-approved gene therapy programs, international long-term follow-up related regulatory guidance, as well as glossary resources. Just to give you a little taste, we'll talk a little bit about a couple of the guiding principles. So the first one, which is actually number 7, this is a big theme throughout the toolkit, which is that there are trade-offs between expanding the scope of long-term follow-up studies and minimizing study burdens on participants, sponsors, and others. The need for long-term follow-up data collection and monitoring should be balanced with the need for participant adherence and retention. The burdens of long-term follow-up studies on participants and study sponsors should be justified by the knowledge to be gained about the benefits and risks of gene therapies and minimized to the extent possible. And after hearing some of the conversations this morning, I would say this sort of aligns with what we heard about minimizing the complexity of studies. Another principle, number 18, and there's, I think, 19, sponsors and researchers should make every effort to publicly and transparently share final and interim as appropriate aggregate results of these studies. So now just to give you a sense of some of the considerations and recommendations, which is really the most detailed section of the toolkit. That section has 9 subsections. By the way, I really think that when it comes out, I'm just going to shout out for the informational design done by Lindsay Henriquez. It's a really fun document to navigate. So even just for that sake, try and open it up and see. So as you can see, there's 9 subsections of the considerations and recommendations, and they really are aligned with different aspects of a protocol and operationalizing the protocol. And so again, just to give you a little flavor, one of the sections is on primary, secondary, and exploratory objectives of the study and corresponding endpoints. And the way the considerations and recommendations section is formatted is that we have considerations and then sub-recommendations under those considerations. So consideration 1 in this instance is the involvement of patients and caregivers is critical for the ethical design and conduct of long-term follow-up studies. Their perspectives on which long-term follow-up data should be collected and how may differ from those of sponsors or regulators. And then we have a recommendation that patients and our patient advocacy organization should be included in the design of long-term follow-up studies to ensure that the endpoints are meaningful and relevant to patients, their families, and care partners. And someone asked me this morning, some of these recommendations are really applicable to all types of studies, right? But we just tried to be as comprehensive as possible in the toolkit. Couple considerations and recommendations related to data sharing and dissemination of results. So the consideration is, at the present time, combining or linking long-term follow-up data sources may result in operational burdens secondary to the lack of interoperability and the high cost of doing so. While it's important to consider the optimal fit-for-purpose design of each long-term follow-up study, it would be scientifically valuable to compare long-term follow-up outcomes in particular diseases or across diseases, and also to compare within and across gene therapy types, classes, or categories. And so the recommendation here is to maximize the scientific value, interpretability, and interoperability of long-term follow-up studies. Endpoints and data collection should be harmonized and standardized to the extent possible to facilitate meta-analysis across products and patient populations. And then one more. The consideration being timely and transparent reporting and publication of interim and final study results maximize the scientific value of these important studies and increase generalizable knowledge about methodologies and results, including awareness about potential safety concerns. And then the recommendation, sponsors and researchers should make every effort to share final and interim as appropriate aggregate long-term follow-up results publicly and transparently, hearkening back to the guiding principles. And then we have a looking forward section, which really just focuses on questions and considerations that are really not completely resolved, so that we want to pose to the field and industry that need further discussion and hopefully moving forward on some of these issues. The looking forward, this is just a few of the issues that we bring up in the looking forward section, but one is data harmonization, which we talked about in the other sections. What incentives, if any, will drive efforts to harmonize data definitions and collection, optimize interoperability, and share data and results to maximize value? We also raised the question of, would a central repository or registry for long-term follow-up data, which might enable studies that include larger numbers of gene therapy recipients, be useful? And if so, who should develop and manage such a repository? And lastly, what incentives, if any, will propel increased long-term follow-up data transparency, information sharing, and reporting of results? And so with that, I'm going to ask our panelists to come up and join me in the chairs. And just to introduce them very briefly, we're really grateful to have Lara Gell from Bristol Myers Squibb, Daniel Kavanaugh from WCG, and Pamela Tennerts from Medable joining us for further discussion.
Hi, everyone. I'm Pam Tennerts. I'm the Chief Medical Officer at Medable, and we are a technology company that helps run decentralized clinical trials. And the reason I was so happy to be part of this working group is because we're really trying to do things differently and making long-term follow-up studies more feasible. And I think that technology will play a big role in that. And so I was really happy to participate and listen to all the thoughtful comments that came for 2 years in a row. I learned a lot. And yeah, that I'll turn it over to Daniel.
Thanks. I'm a senior scientific advisor at WCG. I used to be based here in Boston as an investigator at Harvard Medical School, developing preclinical and clinical gene transfer products and working on a committee that reviews those, the Institutional Biosafety Committee. And now at WCG, we work with hundreds of sites using IBC review services and thousands of sites using IRB review services that get a very wide variety of gene transfer protocols. So we're very interested in the informed consent issues and safety issues.
Hi, everybody. Lara Gell, very happy to be here today. I am the therapeutic area head in global trial management at Bristol Myers Squibb for cell therapy. So my remit is overseeing all phase 1 through 3 cell therapy trials across all of our products and indications. I think what's interesting to me about being here and talking about cell therapy long-term follow-up is in my past, when I was growing up as a clinical trial researcher, I was a trial manager on an open label extension trial in a oncology supportive care. And that was kind of my side job, right? I was enrolling patients into a phase 3 trial, into phase 2 trials, and then those patients would eventually roll into the open label extension. It was a relatively easy thing to get those patients to roll in because we would continue providing product to them in exchange for their data, and they really wanted to continue receiving the drug. And if you look me up, I'm published for 3 times as a result of those annual updates of reporting interim results of that open label extension. That's a very different perspective when I came to BMS and saw that my team was running the long-term follow-up study, and they brought to me and said, we've got all these challenges, and this is a problem, and we need resources, and we need this. And I said, well, gosh, this should be relatively easy. And then they educated me, and it's not. It's very, very challenging.
Yeah, so we're very appreciative of all of you being here with us today, and I thought we would just go around and let you each share a little bit more about your perspectives and highlight different issues that you encounter in your work and roles. So I think, Laura, you're going to start.
Yeah, so sponsors are entrusted with monitoring the safety and the long-term durability of these products, and it comes with unique operational and scientific challenges, especially as the treatments move into new indications, earlier lines of therapy, and actually new therapeutic areas. And so I'm not sharing anything that's outside of the public domain, but the long-term follow-up study in Bristol-Myers Squibb's cell therapy program has all patients from all studies that have ever received a cell therapy in a phase 1, 2, or 3 development project going into one trial. And one of the biggest challenges that we have is that we are constantly adding new trials. A new patient, there were multiple patients that were infused today. And so this trial will now go on for another 15 years from the next time another patient is infused. And so we're in a constant state of startup as we expand our footprint in cell therapy, which is a very exciting thing for us, but it's also very challenging because every time we add a new clinical trial, we add new clinical trial sites in new countries, we need to activate the LTFU to ensure that patients could roll in if they needed to drop out of the parent study, is what we call them. And so that long-term startup is exhausting. I think from our perspective too, Bristol-Myers Squibb acquired a couple companies who had the cell therapies, and as a result, we've got heritage processes and heritage ways of working that were built into our current protocol. We've since worked to scrub out and really simplify the long-term follow-up trial to look at what are the most important things that our products need us to be reporting on and that our regulators need us to be reporting on. And I'd really ask the team to look back and say, if you were to redesign it today, what would it look like? If you were not incorporating something that was designed 5 or 6 years ago, how would we look at this differently? And so what I tell my team is we designed the Maserati of all long-term follow-up clinical trials. And it had all the bells and whistles, and it was expansive. And now we need the smart car. And so and maybe we don't need a smart car, maybe we need a Honda Odyssey minivan or something. But it's thinking about those types of things that, all right, there are a lot of nice to haves, and a lot of scientific questions that we want to answer, but what really is fit for purpose from a operational standpoint for our sites, for the patients who are continuing in this? In some of these cases, the patients have received, well, in all the cases, they've received one treatment, and then they have to come in every once a year, or it previously was a few times per year for the whole 15 years. We've since decreased that down, and after 5 years, they only need a phone call as long as everything is going well for them. So those are some of the ways that we've handled some of our challenges.
That's really helpful. Just a quick follow-up. So one of the things that we did talk as a working group and in the toolkit is about how with such long studies, you need to potentially anticipate the need for protocol changes and flexibility in the protocol. And so I'm wondering, is when you would say to people, how would you do it differently? Is that something that you would also consider, how to build flexibility?
Yeah, it turned into a full protocol amendment and really redesigning, along with our CRO, how we are working with them and how we're changing the way that we're monitoring, but really working with clinical science teams on to try to drive how can we make this more patient-centric from a protocol design standpoint. Yeah.
That's great. Pam, do you want to go next?
Yeah, so what I would bring is that we've had a lot of conversation with customer sponsors who are doing long-term follow-up trials and are really telling us that it's becoming impossible because of the cost and the burden on them, because of some of the things that Laura was talking about. We've heard that a couple of times. The other part is that it's also really hard for participants to participate in this because they're signing up for 15 years. Some of these people come from really far away because you don't get a cell and gene therapy at your doctor normally locally down the street. You get that at a tertiary center. That tertiary center may be in a different country, and you're willing to do that to get that therapy that may change the course of your life. What we're finding is the Emily Whitehead Foundation presented their survey to us, and there's a huge attrition, even from the original clinical trial to the long-term follow-up, and continuing on because it's just hard to do. So there are options with decentralization where you can make some of that a little bit less burdensome on the participant, and technology can also be easily adapted. If you have to make an amendment, you can change it really easily. So you get into scalability, flexibility, and adaptability. We also in the toolkit talk about registries and real-world evidence, and that's then there's like the follow-up studies after the clinical trial, and then there's the approval follow-up studies. And that's where the toolkit elaborates a little bit about how you can think about real-world evidence and things like that, and the use of registries. How can you utilize the use of registries to, again, minimize the attrition that might happen and make it really easy for people to participate, whether those people are because the scalability also goes to the original site. So they do one trial, they have to do they're now going to do the 15-year long-term follow-up. If you do it in a traditional brick and mortar follow-up, if they do the second, the third, that adds to their long-term follow-up studies too. So it's hard for people to do. So if you use a registry, that may make it easier on everybody because the initial trial site may not need to do the responsibilities of being responsible for all the follow-up either. And then lastly, I think you asked me to talk a little about endpoint selections. And I had to ask Carolyn, as an aside, a question, because I'm like, what is the Goldilocks issue? And I guess we did not learn the English fables. We had like La Fontaine and Grimm and all of those, because I was like, I don't think I know what this is. And she's like, porridge. I'm like, anyway, what it means apparently is like getting the fit for purpose, like doing the trial in a way that gives you the answers that you need and make it less burdensome, or only as burdensome as it needs to be for the participants. And that goes completely back to quality by design that Anne-Mika O'Connell, and that we worked on it when I was at Citi, worked on. So this is why, for me, this was a really interesting space, because it is difficult. And at first I thought like, oh, yeah, we're going to do quality by design. But you kind of have to think about it a little bit differently. But it does, it goes back to the same principle. Focus on what you really need on a minimal data set. Get those things right. Make sure that participants agree with that, bring the input of the patients into it. And foundations like Emily Whitehead Foundations are fabulous to do that kind of stuff. There was also, I may stop there because I don't want to.
No, we're
Okay, we have time, all right. So then the need for standardization. I told Barbara I had to chuckle a little bit when at the last sentence of the previous panel was like, well, maybe we can standardize some of these AI elements in informed consent. So standardization is also not a new concept, but really we need to have common data models so that we can start sharing that information, because information on safety, if you're using the same vectors, you can probably put that information together and maybe down the road have to do less monitoring because you know the answer by putting the studies together. And it also goes back to common data elements in all protocols, and then the vertical different ones, like Andy mentioned earlier in the context of feasibility, a specific protocol, if you're doing something around sickle cell or things like that, that may have different endpoints for durability and things like that than maybe an oncology clinical trial where you wouldn't have those endpoints. Remaining issues, I do think we need to, and I think there might be a question about trust later, but what the Emily Whitehead Foundation Executive Director George told us too is that a lot of these participants aren't told at the beginning of their original gene therapy trial that they're in for 15 years of this. And so then when you talk to them about the end of the year, they're like, what? I mean, that's my interpretation. But like kind of like, oh, I didn't realize this. And I think people want to do this. I'm on another project with Citi where one of the patient leads on this project has received a gene therapy. And he said he wanted to participate in the 15-year follow-up because his children have the potential of having the same issue as him. And he wants to make sure that by the time his children might need it, he would be participating and contributing to the evidence, but they were making it really, really hard on him because he had to fly to this center and do all these things. And so I think, this work is so critical in helping us getting to those places where we're a lot more fit for purpose. We can combine data, we can harmonize and do things like that. And I'll just stop there.
Yeah, so that's great. And just to add to what Pam said about the Goldilocks issue, we were using the Goldilocks kind of just as a phrase or a way to talk about like just right, like just right amount of data collection, like what's too much, what's too little, and what's just right. And I guess this is always, again, this would be an issue with any clinical trial or clinical study, but when it's really long, and you really want to minimize burden as much as possible, it becomes a really critical design issue.
Right. And if you start combining data, you can envision situations where Lara just mentioned that they were able to decrease the level of follow-up because they learned that maybe it didn't need to be quite as much. So there's that opportunity to then decrease even further after 5 years or maybe after 10 years or maybe stop eventually. We might not need to go 15 years. If we are careful about how we collect this data, we might be able to make it less burdensome even in the future.
Yeah, and then before we move to Daniel, I just also wanted to ask you one other question, which is there was recently some FDA guidances that were released in cell and gene therapies, one of which was a guidance on post-approval methods to capture safety and efficacy for cell and gene therapy products. And so this was really timely, coming out right around the time that we're going to release the toolkit. So we went back and read the guidance and we referenced it in the toolkit. But I just wanted Pam to speak to that as well.
Yeah, because when that guidance came out, I was like, oh, wow, this is so interesting. It's as if Carolyn knew what was going to be in the guidance and the working group knew. And here are now tools that can help you make that guidance more actionable. So if you've read that guidance, then the toolkit is really a good place to kind of go like, okay, how can we think about that? How can we put those elements in place? So I thought that was really fascinating.
And one of the things that the guidance goes into, which the toolkit also talks about, is using decentralized elements, use of registries, use of real-world data.
Yeah.
So again, trying to minimize the burden of these studies, but also still get the information that is so valuable and still recognize, I think, I haven't heard of anybody saying they're not important. And now we're going to turn to Daniel with that little lead-in.
So one point that's worth making is what is the underlying motivation for these types of follow-ups? When we introduce changes into the chromosome, there's a risk of accidentally deactivating a tumor suppressor gene or activating an oncogene. And then you have a precancerous cell, which may persist for an indeterminate amount of time, for months or years, and then become a clinical malignancy. And we don't know how long that may be with different modalities. So as gene editing becomes more precise, this risk category should decrease, but it'll still be an issue that needs to be thought about and needs to be addressed. Separately, there are other gene therapies that do not modify the chromosome. They introduce extra chromosomal nucleic acids, but they still induce long-term changes, persistent changes. For example, a transgene may be expressed long-term in the liver, and if the immune system decides at some point that that's a foreign non-self antigen, then you can have a CD8-mediated hepatitis. Or if you introduce a gene expressing a clotting factor, For patients with hemophilia, you may develop antibodies against that factor, which are now inhibitors, and that's a very important adverse event that has to be tracked, and we don't know with different gene therapies what the timing of that may be. So those are reasons why regulators specify long-term follow-up after the parent study may be done to track these unknown risks. And that allows IRBs, for example, to really have a good sense of the risks and the benefits at the time of initiation. And that plays into informed consent. So we heard a lot about challenges with informed consent already relating to sometimes simpler modalities. When we're looking at these complexities, which I just mentioned, which is basically just on the surface, how do we explain those challenges to subjects who are enrolling at the beginning of an interventional portion of the study? And then how do we explain to them why someone is going to be following up with them for up to 15 years? And if the regulator says that sponsors must make all effort to try to avoid loss to follow-up, how are subjects motivated to participate without coercion, and how are they encouraged to have a complete understanding of what's going on before they commit? So sometimes the long-term follow-up is integrated into the interventional part of the study. You have a single protocol. Often it's a separate protocol. Even if it's a separate protocol, as just mentioned, best practice should advise participants prior to enrollment in the interventional part that they're going to be expected to participate up to 15 years under current guidances. Or if you have an integrated study, the consent for the long-term follow-up needs to be included at the time of initial consent. In either case, we've been thinking about different ways to reduce the burden on subjects and the cost to sponsors for all of these follow-up issues. And some of those are going to involve real-world data. The trio of FDA guidances that were just reduced, all of them had discussions of real-world data as an important source of information, particularly for FDA-approved products. So there's different interesting new technologies to make that work, to extract data, perhaps through tokenization from electronic health records to minimize the burden on patients and to make things efficient and to get the most feasible and practical information you can without overburdening the system.
Thank you so much for adding those perspectives. I just wanted, you mentioned that the FDA guidance, I think at least one of them, has a phrase that says sponsors should make every effort, is that the phrase? Something along those lines, to minimize or prevent loss to follow-up. And I think that phrase speaks to the importance of long-term follow-up and the regulators' desire to understand the long-term benefit and risk profile of this new class of therapeutics. But as an IRB person, right to withdraw, I'm a bioethicist at heart. So the right to withdraw is obviously a key research right. And so I just was wondering how you mentioned it. So I was wondering how you think sponsors should balance that right to withdraw with this sort of, scientific validity, ethical, we need to. So at the end of the day, if we don't have the participants staying in long-term follow-up, we won't have the data, which this room understands. But so we have this tension again about, having participants complete the long-term follow-up so that we understand and have the data that we need to assess what we're trying to assess. But if it's difficult for the participants to stay in.
Right. The more difficult it is for the patients to stay in, the more likely you're going to run into this exact problem. And then you have to, patients have potential personal benefit from careful monitoring of adverse events, also from understanding of the benefits, the clinical benefits of the ongoing results coming from the clinical trial. Those are things that, reasons for people to potentially want to participate when they're considering consent. And then there's obviously the benefit to the community or to family members who will be affected by the same genetically inherited diseases. And then, of course, the wording also has to be non-coercive and explain to them that, you always have a right to withdraw and you always have a right to refuse to participate. So it's an important balance to try to strike.
Yeah, that's also important. And I just want to add that in the toolkit, we also mentioned that it's important to explain to participants that withdrawal from long-term follow-up does not mean withdrawal from the gene therapy. And so, the benefits that you're mentioning of follow-up, we just have to make sure they understand. Another issue we raise in the toolkit is we raise this whole issue, which I think is a little underdeveloped in the discussions around this, which is, the responsibilities of sponsors for long-term follow-up versus, when and how the responsibilities for follow-up and monitoring transfer to the context of clinical care, and making sure that that really gets, communicated to physicians about reporting adverse events that gene therapy recipients may experience into standard pharmacovigilance mechanisms. So anybody have any other things that they want to add or respond to?
On that note, there is an FDA, I think it was an FDA guidance that came out perhaps last summer where the guidance indicated that patients needed to be followed for their whole life. And I did receive some recommendation from our CRO who said, oh, well, now instead of 15 years, it's a lifetime. And I said, no, my interpretation of this, to your point, is as a sponsor, our requirement is up to the 15 years, and then once they've surpassed that, then it falls into clinical care to track that and make sure that those patients, if they do have a malignancy, that those are reported appropriately.
Yeah. And the burden to sponsors is real because I'm sure, you've seen the news where companies are divesting their cell and gene efforts, and it's been a couple of big ones in the last couple of months. So I think, I'm not saying that the long-term follow-up is the cause of why they're divesting of those things, but it cannot help that if you know that you start on something, the net present value still has to be there for companies to sort of make the decision to move forward. And if people stop working in these cell and gene therapies, that means a whole lot of patients and people that may not get a treatment that could potentially work, because so then we have a lot of diseases that we don't have cures for or even medicines for. So I think anything we can do to make that balance work and have the right amount of data that we need to satisfy, that we can make sure that the safety is there or the durability of the treatment is there, and versus the other things, I think it's just something to really think about.
I can also share the original FDA guidance on long-term follow-up in cell therapy does indicate that sponsors need to make sure that they are financially prepared for the long-term follow-up.
Right.
And it's interesting to me that the FDA calls out the financial preparedness of a sponsor to continue that.
Yeah.
Yeah.
And not to speak of all the, like what we touched upon in the conversations, I'm not sure if it made it in the toolkit, was what if some of these very small companies don't make it, and now you have a set of patients that have received something that's permanently, because you can't withdraw from your therapy in this case, permanently in your body, like how do you deal with that, and should there be some standard mechanism to follow those patients, because clearly the original company isn't there anymore. So there's a whole lot of issues around this. Yeah.
Yeah, so I think just a couple points. And so in terms of the clinical care, transition. In the toolkit, we do, and again, we're putting final touches on it and stuff, but it's going to be released November 4th. But so in the draft of the toolkit, which is being released as a draft for public comment, we do discuss this issue, and we came to a similar conclusion in terms of the interpretation of the post 15 years follow-up. At the same time, we also feel like the guidance isn't crystal clear, and so we do also make that point. In terms of the responsibilities of gene therapy sponsors and developers, the guidance, as you mentioned, is clear that in multiple regions, by the way, I think that there's notes about responsibilities of sponsors about long-term follow-up, and interestingly, just today, a few of my colleagues from the MRCT Center and I have a paper that's out in Gene Therapy on just a very quick analysis of long-term follow-up studies in registered in clinicaltrials.gov. And we did find that, evidence that companies were having challenges for financial reasons and otherwise, and that some long-term follow-up studies were being discontinued. And so in the guidance, it does say that, you should, consult with the regulatory agencies to figure out what to do. And in one of the webinars, we had someone from the FDA come out, and this was in 2024, and they said that they would try to work with the site investigators to continue the long-term follow-up, but of course that also speaks to, how is that funded? How is that possible? But again, when we think about the stakeholder table and about people who are deriving value from long-term follow-up studies, perhaps, we should think about solutions that are, how we can think about making sure that patients are followed up in the context of clinical care, if at the very minimum, right? So, anybody else want to add anything before we go on to some questions? Okay. Laura, I was going to ask you, how are sponsors adapting long-term follow-up strategies to better align with patient needs and real-world feasibility? So this kind of continues our conversation.
Yeah, I mean, if you think about some of the challenges relating to a patient being on a trial for 15 years, they could relocate and therefore not be near their original site. They'd obviously likely change care providers. Patients or pediatric patients would age out of a pediatric program. And really just identifying ways to maintain that patient engagement through the site and be flexible if a patient does need to transfer. And I think one of our trials generally, our parent trials are generally 2 to 5 years in length. And then once a patient completes that parent trial, then they go into the long-term follow-up. And so for those who are on a parent trial that was 5 years in length, and then they go into the long-term follow-up, they actually can start at that point with an annual phone call, assuming that all is good and nothing else has been untoward has been identified clinically. And so that could happen from, Boston to Florida when they move to Florida, right? I think there are other ways that we're looking at being a bit more flexible. So we're looking at tokenization of patients, and I think a variety of different decentralized options. We have looked at like online informed consent in order to ideally get those patients to transition from the parent study to the long-term follow-up more readily, because we don't want them to have 2 separate visits. And one of the important things is we've indicated in our long-term follow-up trial that the first visit should be the last visit of the parent trial. So they are the exact same visit, and you don't have them coming back again the following week to the major health health center for that. So just trying to think really clearly about what would you want your mom or dad to need to do? What would you want to do if you're a young woman of childbearing potential who has lupus? And we've treated you with our CAR T. What are you going to want to do when you go into that long-term follow-up and you've got young kids and you've got a family and you've got a job? And so how can we make it easier so that we can continue to collect that data?
Yeah, that's great. And then Pam, I was wondering if you would just, offer your perspectives on decentralization, how we can bring decentralized elements in, and not just for convenience, but also for sustaining engagement with participants and balancing flexibility with the need for data, as we talked about earlier.
So I think if we talk about engagement, it's sort of engagement drives compliance in some way. Like if we can keep people engaged and they comply, we meet the regulatory requirements. So there's that really having to sustain both is really, and the way to do that is by making the elements that you use as consumer friendly as possible, as easy as possible to do, so that it's not hard to stay involved, because it's a survey on your phone. It could be local visits to your doctor and local labs instead of having to come back to Duke University come back to Boston if you now live in Florida, to sort of do it that way. And I think the other thing is sort of making sure that whatever you have designed had that patient input to begin with, so that you know that you're creating something that people that live with that condition can actually do, because sometimes those people still have other comorbidities that make it hard for them to travel, and it's not just all that, or it's a child and the parents have to come, all those things. So I think it's really minimizing burden by flexible design, and then just staying on top of regulatory integrity and oversight. And as the guidances change, you should change along with them. So when they go from no longer 10 years, maybe a couple of years from now, and it's 5 or 10, 15, it's no longer 15 and now it's 10 or 5, that you can change your device, your trial to keep up with that. But I think tracking compliance. And the other thing is people are saying the right to withdraw, maybe thinking about, okay, maybe don't come back twice a year. Can we do something in between? Maybe we just do a phone call instead of like having you fly to Boston. Why don't we do phone calls instead? And then maybe you don't completely lose the patient. The patient is still tracked for safety, but there's sort of an interim solution. So I think we can be a little bit creative. And I think if you explain all that, I think the regulators would prefer a phone call than nothing at all. So I think there's options that way as well.
And so an important part of designing those approaches is that you still need to be able to recall people after an important adverse event.
Right, correct.
Particularly malignancy. People who have experienced cancer are at risk of recurrence, unrelated to treatment. But a chromosome modifying modality will leave a genetic footprint that can usually be identified in new malignancies that help establish causality. That's the easiest one. A lot of these other questions of causality for adverse events are more complex, but they still need to be addressed. So a two-step process of
Correct.
monitoring, checking in, but also maintaining the ability to get follow-up biopsies, et cetera, when needed for causality is important.
Yeah, so Daniel, I just wanted to follow up on that with you, which is, as information and knowledge grows, as the study progresses, participants may need to be updated, right, on new information about the benefit-risk profile of the product. Now, again, they can't necessarily stop taking the gene therapy, but they may be, there may be a need for increased monitoring, perhaps decreased monitoring, and there may be potential changes to the protocol, as Laura and Pam have suggested. And so I'm just, wondering if you could share with us how best practices around how participants should be updated about this new information.
Sure. And I think what we heard this morning is that informed consent is a process, but sometimes it is presented as a document, and that is something that clearly does not work for most of these trials. It has to be a dynamic process. And one example is that a whole class of gene therapies are used with very young children or neonates, and in the world of rare disease and ultra rare disease. So those treatments will be administered via parental permission, excuse me, and then assent will be applied as age appropriate as the children grow older, and that includes, assent to participate in long-term follow-up. So there's an ongoing challenge to keep that population engaged and make sure that assent and permission are properly managed. In addition, for anyone who's involved in these studies at any age or competency, there's an ongoing fact that over time there's changing information. So, and if the information actually would affect a person's willingness to continue participating, that should go into an informed consent update. Other new aggregate information about safety and efficacy also goes into these regular updates and then amendments to the protocol. So it's quite a dynamic consideration over time. And, listening to speakers this morning, I was thinking of different ways to make that more user-friendly using some of the technologies that were suggested, but also keeping in mind some of the barriers, the difficulties to implementing that we heard about. But obviously, this could be a fruitful area to introduce new technologies for keeping people informed and engaged in these trials with ongoing consent.
Yeah, so I think that's interesting, and that's, being part of this project, it made me have a greater appreciation for, I guess, the use of updated informed consents to convey different information to participants who, are not being really asked if they want to continue in the study. They're at, the assumption is that they will continue because they've consented, and there's reason, value in their continuation, but also to share information, which I thought was interesting, which I didn't appreciate before having these discussions and developing the toolkit.
Yeah, the amended informed consent is one tool for updating participants on the latest safety data and sometimes efficacy data.
Okay. Are there any other tools that are used to update participants if they're in that case?
So I wouldn't say specifically within a long-term follow-up trial, but once you finish the parent trial, and sometimes there's information that goes back to the patient from the physician on, the trial was positive, or it read out, or the drug is now approved, right? And so if there were a way to make sure that that's happening on an ongoing basis and I don't recall if it is specifically happening once we finish the parent trials.
Okay, and I just want to mention, so in the toolkit, which is again coming out November 4th, but we ended up including a section, as I mentioned in the intro, types of long-term follow-up studies. And part of that was because as we dug into this work, you probably also might remember flow charts. So part of the resources in the toolkit are really just sort of enabling us all to get our hands around the complexities of just this one specific category of clinical studies. Because as Daniel mentioned, there's integrated protocols. In other words, the long-term follow-up is integrated into the parent investigational trial, or they could be standalone. There's benefits. There's advantages and disadvantages to both designs. We also, had discussions about observational versus interventional, those classifications of studies, and how that impacts reporting requirements for the studies. So in other words, if we say as a guiding principle that we think results should be reported, how they're categorized, whether they're interventional, in other words, an integrated protocol would be interventional by definition because you would have the parent trial. But if they're standalone, there's a lot of detail into figuring out, and I'm not a lawyer, but into figuring out whether they would be classified as observational or interventional, and that has implications for reporting requirements and registration requirements. So it was just interesting to get into that level of detail.
Yeah, I can say that ours is an interventional trial because there is prospective testing that is required at certain time points throughout, right? And so part of that, as I mentioned, we have to have the site set up in order for a patient to roll in, but we also have the central lab set up. We also have additional, systems available for when those patients come in and the testing that needs to be done for them. And so, unfortunately, in a standard development program, it can't be observational only, right? Based on the guidance.
Oh, can we just wait for the microphone? And yes, it's time for questions from the audience, so that would be wonderful.
So I just had a question for you about that. So it's an interventional trial for your long-term follow-up. What kind of milestone cuts do you take to report it out? And do you do an actual CSR and report it out on ct.gov, those aggregate data for just maybe even just the safety, or how do you handle that reporting?
Yeah, so we're doing annual data cuts that's reported to the FDA, but we haven't published or put the results on ct.gov because the trial is still ongoing and patients are still being recruited.
No, but I mean long-term follow-up.
Yeah.
So, but they're still being recruited, but it's always going to be
Right,
recruited.
right, yeah.
So then
So
you would never
at this
put them
point,
out?
we have not done that.
And so just to say, I'm just going to add that, we did find that, in discussions, this does seem to be an issue with long-term follow-up studies, that they're not necessarily being reported, partially because of their length, right? Normally, studies are reported at the end of the study, right? So there's a little bit of that as well, which is, but the interim results would have value. So that's one of the, recommendations that we have in the toolkit, which is how do we increase sharing of results on the one, and then also even sharing of data, potentially in data platforms, which again, it would be probably a higher bar, but still we can be aspirational.
Hi, what are the panel's thoughts on this? Every once in a while we see a story about how post-marketing studies just aren't being done in accelerated approval situations. If a long-term follow-up is analogous to that for cell and gene therapies, what incentive do you think FDA might put onto cell and gene therapy companies such that the toolkit will actually be used?
I think that's a great question. I think, first of all, I think post-marketing studies are different, right? So there's an approved product. I think now in some types of long-term follow-up studies, that could also be the case. I think that there is a recognition that we need to do these studies. There's still remaining uncertainty. That being said, there is a recent white paper that was led by Catalyst and included Emily Whitehead Foundation and Friends of Cancer Research and some representatives from industry sponsors who have recently proposed that long-term follow-up studies for CAR T products be reduced from the recommended 15 years to 5 years. So, and that's available online. It's not a publication as far as I know yet. I think it has been submitted for peer review, but I'm not sure if it's actually been published yet. But you can get the white paper. But so I don't know in terms of incentivizing. I think that the FDA guidance is pretty clear that they want these studies done. But what I think, it's a great question. Because we do know that then some of them have been discontinued, and we do know that this field is under strain and stress, which is perhaps reflected by the FDA guidance, is trying to make it clearer and show that they're open to, trying to support the development of this class of products. So anybody else?
Yeah.
I guess that's a non-answer, but bottom line.
Well, I think your question is relating to transitioning from accelerated approval to full approval. Is that right? Yeah. Right. And so I'll say in my experience, the LTFU is not part of that conditional approval. It's already expected that we're doing the LTFU. The conditional approval transition to full approval is either providing longer term follow-up within a parent trial, in which case we'd ideally not have started transitioning patients into the LTFU, or perhaps a randomized controlled trial that has to be well underway at the time of FDA decision. So that is evolving over time and has evolved over the past couple years that I've been experiencing this type of thing. But yeah.
I guess I can add that we've heard recent comments from the FDA about a pathway based on plausible mechanism of action, which would apply to rare, probably in ultra-rare conditions. So a lot of those will be cell and gene therapies, probably. And then the question is, how will those approvals be treated as in terms of the durability of the approval and actionability over years and long-term follow-up for those. And I think we just have to wait and see how those work out. Yeah.
Thank you so much for all your comments. I think we can learn from some other areas as well where there's more populous sort of treated patients. So if you think of something like immunotherapy and a product like Keytruda, which has now millions of patients dosed in that, in the clinical trial program, you'd finish the endpoints, and sometimes you have multiple endpoints like disease-free survival, and then you go for overall survival, and then people want to know what happens then. And so those all went into a single omnibus protocol. So they dumped all the patients from the hundreds of clinical trials into one protocol, and they follow them up. The kind of principles one adopted there was keep it as simple as possible, keep the database design as simple as possible. And the fundamental question is, are they dead or are they alive? So you want to know further survival from that, and so you can get some sort of Kaplan-Meier. And for those who are alive, have they had any serious adverse event? And those who died, did they die of something weird and wonderful? Or was it just progression of disease? Did they develop, and people laugh, but the weird and wonderful, do they suddenly have clotting propensity, or do they have something else that is a warning that comes out, a signal? So that's one way of just looking at what they did. And then those studies typically were not monitored like you monitor other studies. You don't go and verify every bit of source data, especially the survival data. You have to have a certain level of trust in this because you're down to fewer patients per site. And the other thing then is, can patients then transfer, and can you make this simpler? And the type of thing I'm thinking about is what happened in rare disease programs when I worked at Genzyme some time ago was you set up registries. And registry, I don't know whether you're differentiating between registry and a classic clinical trial database as you follow these up, but the registries are sort of very light touch database, and it's not always done at the primary treating physician. You can have an e-access into a web base that people can impute, a GP can impute. And so I think you have to look at the follow-up care of over 15 years. Who's actually seeing these patients? And is it always the primary physician who saw them to treat their disease initially, or are they now in the community base? And that may determine what you're doing. And I agree with Pam's sort of comment there is, can you make this sort of an annual sort of check-in and keep it very simple in that database, and that way you can follow up. And then I think the last comment was a really important one, is what happens in the small company scenario where the company may go defunct and everything, and you still want to get follow-up. So I think there should be some recommendation of having a patient profile drawn up that describes this, and the patient can then take that, and it can be given to their next treating physician, whether it's a GP or a specialist or whatever it is, that they're aware of this, and there may be some spontaneous reporting, if not to the sponsor because they're defunct, at least into a regulatory agency, and there may be some central way of tracking what's happened to these patients longer term so we can learn from those and they're not lost in the sort of shuffle there.
So I'm going to pull in another project that I'm involved with at MRCT, which is the PI oversight, and sort of different scenario, but to your point, Andy, somewhat similar in that when in a regular clinical trial, the patient is being seen by the local HCP. And we have there created a information sheet for the HCP to kind of say this patient is in, because they, per definition, they don't need to be a sub-investigator. They don't need to have protocol knowledge or investigator brochure knowledge. So they just need general information. And it's sort of, this patient is in this trial. He's about, or they're about here in their study. If you see this, give us a call, kind of a thing. So I wonder if that could somehow be applied to this eventually, because it kind of sounds like a similar scenario. Because they need to be informed of something, right? They need to kind of know, here's what the patient had, here's what you might want to look for, you know, and if you see something, call us. And that, and then the check-in would be, if the patient says there nothing happened, then you don't need to check in. But if then something did happen and they say, you know, I had an x-ray and they told me I had something and I was going to my HCP, you could give that information sheet and have the HCP or the local provider look at it instead of coming back to the, because I do think it's a phone call alone. If everything's great, that's fine. But if something happened, you probably do need to explore that further, and that could be a mechanism.
Yeah, we have explored that, a similar model looking at using kind of a centralized investigator.
Right.
So one investigator on the whole of the East Coast, for example, and probably one in the West. And they call the site, and then if they say everything's great, then everything's great. If they say, oh yeah, actually I was recently diagnosed with lymphoma, well, gosh, let's get
Right.
you back into the clinical trial site. We haven't implemented that, but it's definitely a consideration. To your point about clinical monitoring, we had already, when I joined BMS, we were already doing a risk-based monitoring approach. We have considerably dropped that down to a very, very low on-site visits and basically a 0% SDV.
So one of the suggestions that has come up a couple times here is registries, which may combine different populations. And you get into the lumping versus splitting problem. If I developed an editing product with what I think is the best guide RNA that is most precise and is not going to give me, is going to minimize off targets, I may not want to have that lumped in with someone else's product. If I think that's going to have more off target indels and a greater risk. So you want to consider that as, you know, when you're planning, how do you developers minimize the risk of getting mixed in with the wrong population in terms of adverse events. But obviously, combining things together has a lot of advantages for getting a big picture of outcomes and safety.
And I will say that registries, as you've heard this afternoon, came up, right? Registries came up, and the use of registries in how that relates to long-term follow-up for gene therapies. And so we had to dig into registries and, you know, how they are being used and distinguishing that from registry-based studies. And then, again, figuring out, you know, how and when they are used with respect to long-term follow-up. There are a couple examples of registries. And in fact, in the Innovative Approaches webinar that's on demand, we had, there's the CIBMTR registry for CAR-Ts, Center for International Bone Marrow Transplant.
Yeah.
If I got that acronym right. And then the World Federation of Hemophilia has a gene therapy registry. So that webinar actually goes into 2 existing registries for long-term follow-up of gene therapies. Not all long-term follow-up post-approval studies are registry-based studies. Some of them are just more typical studies that don't employ or leverage a registry. But this is all to say it's in the toolkit because this is where the work has led us. And then what we did was try to share our learnings as we went in the toolkit with others who, you know, for example, small biotechs especially, or academic investigators, or anyone else who gets themselves involved in long-term follow-up, whether by, you know, professional role or even potentially as a patient, then you can find out a lot of information and again, ideas about best practices.
Can I mention something real quick that I don't think we've talked about that's in the toolkit is the master protocol approach, and that is maybe more a scenario that would work in your case, where if you have a master protocol and every company has their own little arm, then that's where you can stay separated from some of those getting lumped in with everybody else's adverse events and things like that too. So there's also a description of potentially how to think about master protocols to get you the information you need. And
Yeah, and that's also that we also discussed that in the Innovative Approaches webinar a bit too. So all this to say, so we're coming up on time, but I want to first of all thank our panelists for traveling here and being with us today and all their contributions. And just to sort of pitch one more time, our toolkit is being released on November 4th. We will have a webinar that day to mark that release. And I just want to again express all my appreciation to the working group members and to the MRCT Center Cell and Gene Therapies team. And by the way, we also have a strategic advisory committee for the Cell and Gene Therapies project. So thank you to those folks as well. So that's it.